Good evening, geneticists. We've made it to week 15. Wow, this is the last week of instruction, and I owe you a video on eukaryotic gene regulation. This is barely going to scratch the surface of this complex topic, and I'll be honest with you, I'm sort of distracting myself by doing this. It's a little hard to concentrate with national events as of late, but here we go. Eukaryotic gene regulation. Let's do it. So, you have already seen prokaryotic gene regulation in the form of operons. And what you'll notice here with this eukaryotic gene region is that all this white area is non-coding DNA. There's a heck of a lot more non-coding DNA in eukaryotes than there is in prokaryotes. Our genomes are way bigger, and a lot of that genome is not ever going to code for a protein directly. Instead, a lot of what that non-coding DNA does is modify the transcription and translation of the coding genes. So in this segment, we have a blue coding sequence, the CDS at the 3' end. All the white is non-coding DNA. Upstream region, anything to the left or 5' of the +1 site in our initiator element is considered upstream. That +1 site is where we're going to orient ourselves. Anything to the right or 3' of that +1 is considered downstream. Oh wow, I just threw a bunch of stuff on here. So all these green regions are promoters. So remember back to the simple days of the operon, right? There was one promoter. Here you've got 1, 2, 3, 4, 5, 6, 7 of them. And all of these are working in concert to increase the chance that this gene gets transcribed. If you mutate one or more promoters, the rate of transcription goes down. That's how these expression studies were first done in the first place, right? You make a mutant, you see what the effect is in the phenotype. In this case, the amount that CDS gets transcribed goes down. Some of these are consistently found in eukaryotic promoters. What I've drawn here is a schematic of a made-up, what's called a focused promoter, which means all these promoter elements are found cis to each other. I'm showing you them in cis arrangement because it's simple, and they'll flow in some sort of what seems to be a logical order. But I want to also throw in the caveat that all of these promoter regions can be acting in trans potentially. So we have things that you see all the time in eukaryote genetics, such as Tata box and the cat box and the GC box. And notice that a lot of these are found upstream or left of the +1 site, such as the Tata box. And then you have some, such as the motif 10 elements and the downstream promoter, which are downstream or right of the +1. So if the promoters increase the chance that a gene gets transcribed, we can really ramp that up with things called enhancers. And this is sort of like an overdrive. If this gene needs to be transcribed in huge amounts above a baseline level, there can be one or more enhancers that increase transcription. And what I've drawn here is just a schematic again, but we have what's called a proximal enhancer. It's located near enough to the gene region, right? In this case, it's at position -90 arbitrarily between a couple of promoters. I've put one at -220. That's distant. This distant enhancer could actually be located thousands of base pairs away. This is just, to fit in the screen. But yeah, you can have promoters extremely far from your coding sequence and also on trans-affecting elements, such as other chromosomes. And if we have ways to increase transcription, it would make sense to have ways to decrease them. And one of those would be a silencer. This one I've just thrown in between a promoter and an enhancer, and arbitrarily at position 160. This would decrease the chance that our coding sequence gets transcribed. This could actually also silence the gene in some tissues versus others, right? Remember that all of your tissues, every cell in those tissues has the same genome, except different genes are expressed in different tissues. That's why those cells have diverged. Okay, so let's say that our coding region does get transcribed. So we'll zoom in on that CDS, and I've just trimmed off the upstream region at this point. What I've broken the gene up into here are these colored regions called introns and exons. We didn't get to this earlier in the class, so Now I'll spell it out for you. But our +1 site initiates transcription. +1, that little arrow there is a standard notation for transcription in expression studies. And so we have that entire area transcribed. The whole region is faithfully copied into mRNA. And the orange are introns. And so there's the yellow-orange color. Introns are removed from a mature mRNA. So they are transcribed initially, but then they're spliced out by a complex of enzymes called a spliceosome. And so what we have here are the chance to actually create alternatives of an mRNA. And this is called alternative splicing. So we're going to remove all the introns. That's clear. Okay, all the orange areas are removed. But here, notice I've got a protein product which consists of, or not protein product, but an mRNA, which consists of exon 1, 2, and 4. I've actually spliced out number 3 between 2 introns. What I could do is I could also make an alternative product that has exon 1, 3, and 4, and I can skip over 2 between 2 introns. These are called alternative exons, and it enables the same gene region to produce 2 or more protein products. So one of the greatest findings, most important findings of the Human Genome Project was we have way too few genes. This is one reason why we can get away with having that few genes. So we have, let's round off to 20,000 genes. It's known that the human proteome probably exceeds an order of magnitude greater than that, around 200,000 proteins. And this is how one way you can get multiple protein products out of the same gene region. Very neat. So let's say either one of these 2 alternative splices will do. We'll take that one and we'll mature our mRNA. So we're going to add a 5' cap to it and a poly A tail. And that superscript N on the poly A tail would normally be around 200. So quite a few adenosine residues are added to the end. So we have gene expression now. We have mRNAs that are ready to be translated. So one thing that separates eukaryotes from prokaryotes, of course a principal difference is that eukaryotes have a nucleus. All the ribosomes and the sites of translation are actually out of the nucleus in the cytoplasm. So mRNAs in a eukaryote are made in the nucleus, and they have to then pass through the nuclear membrane to the cytoplasm. They cannot do that unless they have the 5' cap and the poly A tail. And so then they're translated like they should be, but you can also then imagine that maybe there could be overexpression of a gene just from the mRNA sitting around too long. So there have to be mechanisms that destroy mRNA so it's not just lying around getting translated, wasting time and energy, right? So one way we can do that is deadenylation dependent decay. So we're going to put an enzyme in here, deadenylase, which is going to do as its name suggests, and remove adenine residues from the 3' end. The poly A tail not only transports or allows this mRNA to be transported out of the nucleus, but it allows it to remain in the cytoplasm long enough for it to be needed. But all this time, a deadenylase is eating away at it like a fuse burning down. Once that fuse burns down to the end, we can then have an enzyme called an exoribonuclease come in and start to digest the mRNA itself and get rid of it, breaking it up into its constituent ribonucleotides. There's also deadenylation independent decay. An enzyme called an endoribonuclease, endo meaning within, it's going to cut within the mRNA, is going to separate our mRNA in half like this. And now there's a new 3', a new 5' end that are not protected by a poly A tail and or a 5' cap. And so once again, we have our exonucleases coming in and really getting to work on it in a quick way, digesting, in this case, from the 3' end in that direction. And there could also be a counterpart which would digest from the 5' end, that remaining piece. And this ensures that mRNA is not lying around longer than it needs to be. There are other mechanisms. This is just one set of possibilities here. I'll refer to chapter 16 as well, which has further information on eukaryotic gene regulation. Everything they do is with examples because there's no real set of ground rules here. That's what makes it complicated. If you're studying your favorite gene, the particular regulation mechanisms are going to be peculiar to that gene, and there really are a lot of nuances. And with that, I bid you good night.